Let's start this segment by talking about your biochemical test handout. I forgot to mention that there's a couple of lines that we're going to cross out here. So this first top line of MSA, salt, growth, no growth, you can cross that out. You won't have to know that for the quiz or the lab practical. This KIA, this first line of KIA, glucose and lactose, fermentation enzymes, organic acids, you can cross that out. I didn't talk about that in that segment because we didn't see it in the test tubes that I grew. Some of your bacteria would have grown and changed the media a couple of different colors because there's phenol red in that media as well as those iron ions. And if your bacteria could ferment the glucose or the lactose, it would have created organic acids and turned your test tubes yellow or half yellow and half magenta or all magenta, depending on whether it could ferment those sugars or not. But we didn't see that in the 2 test tubes that I grew. And it's not something that we would have tested with your unknown anyway. So we're just going to cross that out and don't worry about that. You'll have to know this second line for KIA. But what we're going to go over now is this mannitol salt agar. So here are the test tubes for mannitol salt agar. The test tube on the left is the uninoculated control, which means that's the original color it was. It's a red color because phenol red is the indicator in here that we're testing. The middle one is the positive control, and the one on the right is the negative control that was inoculated. So mannitol salt agar is known as a special purpose complex media, and we're just going to write some stuff out here to help you understand this. So it's a special purpose complex media, special purpose complex media. And there are 3 different types of special purpose complex media. There's enrichment, differential, and selective. And you learned about this in lecture quite a bit ago, but now we'll get to see it in real life. And I really wish you could have seen it in real life. So we will do this. We would have done this with our unknowns, and then we would also have done a skin swab to see what kind of bacteria we could pick up from our skin and if it would change it a different color. But anyway, there's 3 different kinds of special purpose complex media. Mannitol salt agar happens to be 2 of these. It's both differential and selective. So it's got mannitol, which we know is a sugar, and it's got salt. So that salt is what's called the selective ingredient. It can select out for bacteria that can or can't grow in high salt. It's got like 5% salt in there, and not a lot of bacteria can grow in a high salt environment. And then if they can grow in a high salt environment, it's differential based on the color it's going to change. So the selective ingredient is the salt. And then if it grows, we're going to look to see what color does it change. So then we would look to see, does it turn it yellow or does it turn it magenta? So let's write out the chemistry here before we move on. So with MSA, the substrate here is mannitol. This is going to look very similar to the Durham tube tests. So the mannitol is the sugar that's in here. The enzyme we're testing for are fermentation enzymes. If your bacteria have fermentation enzymes, it'll use that mannitol and create organic acids. And then the indicator there is the phenol red. So again, we know phenol red is a pH indicator. We know it indicates pH. Red is neutral, yellow is acid, and magenta is alkaline. So if we're looking for organic acids as a byproduct of fermentation enzymes, we expect for the test tube to be what color? Yellow, yes. So that middle one, again, is the positive color. That magenta one on the right-hand side, that bacteria went through some other metabolic process. It could grow on the media, but it went through some other metabolic process and created an alkaline byproduct. So some of your bacteria won't have grown in this media because of the high salt. And then if it did, we would look to see whether it turned it yellow or whether it turned it magenta to see if it can grow, does it ferment this mannitol, and does it create organic acids as a byproduct. So if it can grow in the mannitol salt agar, that means it can handle a high salt. So the selective portion of this special purpose complex media is the salt, is the ingredient that makes it selective. And then the differential portion technically is the phenol red, because the phenol red lets us know whether your bacteria can ferment the mannitol and create organic acids as a byproduct. So the pH, or not the pH, I'm sorry, the phenol red is the differential portion of the media itself. So you're going to have to know this. You're going to have to know that mannitol salt agar is both selective and differential, why it's selective and why it's differential. So we know it's selective because it can handle high salt, and we know it's differential because of the phenol red. So once it grows, does it turn yellow? Do organic acids, or are organic acids present in the media? So one type of bacteria that we know can grow in high salt is skin bacteria, right? Because when you sweat, what do you sweat out? You sweat out salt. So we know that staph species can grow in this media. There's other species that can grow in this media too, but specifically staph species can grow. And then once we know that staph species can grow, if you're trying to determine whether you have staph epidermidis, which is a normal flora for everybody, or staph aureus, which can be a normal flora for some people and is also implicated in infection usually, you can do a swab of this mannitol salt agar, and if it turns it yellow, you know that staph aureus is present. And if it turns it magenta, you know that staph epidermidis is present. That's specifically if we're trying to grow skin bacteria. So if we were in class, we would have made a swab of our skin somewhere, like in our belly button, behind our ear, between our toes, maybe in a place where you have eczema or something. And if it would have grown on the media and it would have turned it yellow, we can make some presumptions that we've picked up staph aureus there. If it would have grown and turned it magenta, we could have made some presumptions that we picked up staph epidermidis there. So if you're wondering about a skin infection, you can use this media to figure that out because it is a selective and differential. And you're going specifically for skin bacteria that you know can grow in high salt. And then from there, it differentiates based on a color change what two types of staph are usually growing on people's skin. Again, this is a normal flora bacteria, staph epidermidis. Everyone's got it. It's usually not implicated in skin infections or anything like that unless you're immunocompromised. Staph aureus, however, some people naturally carry it, and it can be implicated in infection. Usually when you have a scab or something, often it could be Staph aureus that's causing that, a little bit of that scabbing, plus other bacteria. So you could try to determine which one you have based on the color change, and that color change then is the differential portion of this media. So when I do this for class, I usually have you all take a plate and divide it in half and do half a skin swab and half of what's called a fomite swab. And probably more of you are familiar with the word fomite now. A fomite is an inanimate object that people touch a lot. So we would have done a swab of places that you think people touch a lot and see if we could pick up any skin bacteria and see what color it changes. And then you would have done a swab of your body somewhere on your skin and see if we can get the bacteria to grow and what color it changes. And then we can, again, indicate the differential portion because of the color change, whether we picked up maybe Staph aureus or Staph epidermidis. But again, these aren't the only two bacteria that can grow. There are other bacteria that can, and in some cases, your unknowns could have grown in this and turned it a different color. It's just specifically staph species can grow, and we can indicate two of the many different staphs based on the color change that we see here.